welcome Professor Gobetti. Professor Gobetti is joining us from Italy, from the University of Bolzano, and for his impressive bio, please look at the chat. Kim will link that in. Thank you for joining us, and he will be speaking to us about why fermenting both grains and legumes. Thank you very much for being with us.
So first of all, thank you very much for inviting me to this webinar and to the Grain School. It's a great pleasure for me to speak about fermentation, and especially about the sourdough fermentation as a tool for combining grains and legumes. First of all, probably all of you know the sourdough, but when an accepted definition, at least scientifically speaking, definition of sourdough. Sourdough is a mixture of flour. You can make the sourdough using wheat, rye, rice, and legumes, of course, and water fermented by lactic acid bacteria and yeasts, which are responsible essentially for the capability to leaven, especially yeast, while contemporarily and unavoidably acidifying it, and this is the role of lactic acid bacteria. Sourdough, as you know, is one of the last example of natural starters, and I have to say that I started almost 30 years ago to work on sourdough when pioneer studies were already present, especially from Spain, Germany, and Japan. And a number of publications deal with the bread from San Francisco. We have to know that the microbes that are present in the sourdough, essentially lactic acid bacteria and yeast, have two main sources of origin. The first one is from the so-called house microbiota, which means that the bakery equipment, the baker are always sources for decontamination by lactic acid bacteria and yeast, both dominant and subdominant species. The other origin of lactic acid bacteria is obviously the flour, but considering the milling process, recent researches show that most of the lactic acid bacteria are plant endophytic, are representatives of the plant endophytic population of cereals, living with the plant from the first day, from seed to the last day of living in the field. And therefore, they are coming in the flour. Just recently, 1 month ago, we published these articles in Trends in Food Science and Technology, which was a systematic review dealing with 1,300 articles that came in the literature the last 30 years. What surprised me was the following. If you have a look on the sourdough, you can understand that the sourdough has been studied and obviously more used in all 5 continents with a lot of studies, as I told you, more than about 1,500 from all continents and so many countries. So indeed, sourdough is the tool to get the fermentation of, we can say, almost all the species of cereals, all the species of pseudo-cereals that we know, but interestingly, also a large part of legumes. A direct fermentation with sourdough of legumes, including chickpea, grasspea, and so on, faba bean, lentils, beans, lupins. But interesting, sourdough is used also for fermenting agro-food byproducts like with germ, as we had before, with grain, and miscellaneous products. When you want to use the sourdough, all the literatures say that the most appropriate time for fermenting with sourdough is about hours. The temperature should be between 30 and 35 degrees centigrade. And the number of lactic acid bacteria are those one, about log 8.5, and the average cell density of yeast is about 7, with a ratio between lactic acid bacteria and yeast, which corresponds to about 1 to 10, 1 to 100. There are a number of biochemical parameters that you may determine to see if your sourdough is working well or not. In most of the cases, after the sourdough fermentation, your dough, apart from the use of cereals, pseudo-cereals or legumes, should have a value of pH less than 4.5 and a ratio between lactic acid and acetic acid, a molar ratio below 4, which means that to get a nice sourdough, you need a great concentration of acetic acid. Other parameters that you can determine are the concentration of free amino acids as the result of the degradation of proteins, especially by lactic acid bacteria, and the content of ethanol that is mainly produced by yeasts. This is a list of the starter species that can be used to ferment the sourdough. And this is a list of the selection criteria that you can use to ferment the sourdough. And in most of the cases, the secret is to use microorganisms that are coming from the same flours, grains, cereals, pseudo-cereals, or legumes, where you have to use the sourdough. This is just an explanatory graph showing, depending on the size of the name, the most abundant presence in the sourdoughs that you can find worldwide. And these are the species. You have to think that because of the large diversity, because each bakery has its own sourdough, at the end of the day, all sourdoughs are different, but the species that you can find more frequently are those with the highest size in this graph, like Lactobacillus plantarum, brevis, sanfranciscensis, and so on. The same figure could be has been done for yeast. One of the main yeasts that you can find in the sourdough is Saccharomyces cerevisiae, but please pay attention, Saccharomyces cerevisiae, the wild strain, and not as contaminant from baker's yeast. When you ferment with the sourdough, apart from the flour, you certainly have a typical sensory properties. A typical sensory analysis describe the products fermented with sourdough as acidic, with the high activeness, with the crust fruitiness, with the flavor intensity, with freshness, with high porosity regarding especially the crumb, roasted sourness, and so on. There are a number of descriptors that can be found to qualify the activity and the fermentation by the sourdough. Why this sensory profile? Because this is a list of a number of chemical compounds that you can find in the bread or other baked goods which are made with a sourdough, which are strictly related to the species that you can find in the sourdough. So microbes during sourdough fermentation, in particular lactic acid bacteria, synthesize these chemical compounds or synthesize precursors that can be the chemical compounds from which during baking you can get volatile compounds that qualify your bread, your baked good. Legumes. Legumes, as we know, are the second most popular food crop worldwide. Legumes are essential components for the daily diet, providing proteins, dietary fibers, vitamins, minerals, oligosaccharides, and phenolics. And the frequent consumption of legumes decreases the risk of a number of pathologies. I don't want to deepen this aspect. I'm just a food microbiologist. Nevertheless, what we have to say is that the consumption of legumes as they are is too low. Therefore, we have to find a way to increase the consumption of legumes. And legumes themselves contain anti-nutritional factors, which in some cases may give some problem. So the current strategy is to integrate legume flours, to combine legume flours with grains, with cereals, with pseudo cereals, both for making leavened baked goods, but also I have to say for making pasta. Indeed, this is a study dealing, in Italy we have a lot of legumes. In particular, this is a study dealing with the 19 traditional Italian legumes having a specific product, a specific certification for their manufacture. And therefore, these are the main species that we use. So, and therefore, we treat these flowers as we are currently doing with cereals. So we use the legumes to be fermented by sourdough fermentation. And what happened after sourdough fermentation? After sourdough fermentation of the legume flours, if you make a comparison with respect to non-fermented flour, what you can get through the fermentation, you can get an increase of the content of free amino acids, which has a nutritional repercussion, a digestibility repercussion, also including some amino acid derivative like the gamma aminobutyric acid that has also behind the nutrition a physiological role. You may get in legume flours after fermentation an increase of the soluble fibers, and you may get an increase of phenols and of the antioxidant activity which is related to the consumption of legume. On the opposite, on the other side, before I was speaking about the presence of anti-nutritional factor in legumes. Through the sourdough fermentation, you can get, for instance, a great decrease by about 70%, depending on the species of legumes, of the raffinose, which is one of the main carbohydrate giving some problem during our digestion. At the same time, other anti-nutritional factors like condensed tannins may decrease. And last but not least, you can get a decrease of the phytase activity, which in other words has a repercussion on the mineral bioavailability during digestion. So what you can do, you can get a fermentation of legume flours, you can use the fermented dough as it is, or you can mix these fermented legume flours together with, for instance, cereal flours for getting a combination for making bread, but also other type of baked goods, keeping, because of the sourdough fermentation, the sensory properties which are related to products only made by cereals and very well accepted by consumers. This is the case of the sourdough fermentation and Vicia faba flour. you know that in Vicia faba there are two chemicals, in particular visin and convisin, which are responsible for favism. So a part of the population cannot use Vicia faba because of these pathologies due to these two chemical compounds. Then sourdough fermentation, also in this case, you can see this graph, completely eliminated the presence of these anti-nutritional compounds. So the message for increasing the use of legumes is please submit this legume flours to sourdough fermentation, then use them alone or mix with the grains, and at the end you can get an increase of the nutritional properties, an increase of the consumption of legumes, thus respecting the main dietary recommendation. Speaking about other flours, we did some work also on hemp flour, which has a very nice nutritional profile. And also for this flour, we got an increase of the digestibility of proteins, an increase of the dietary fibers, of specific amino acids, which in turn make the digestibility better, as well an increase of fatty acid, unsaturated fatty acids, and a decrease of also in this case anti-nutritional factors which are present also in hemp flour. A few words concluding my speech on key nutritional attributes as influenced by sourdough fermentation. Also in this case, the figure refer to the size of the nutritional attributes, refer to the number of studies that you can find in the literature. And I want to spend just 2 words for the takeaway message I want to send you regarding the sourdough fermentation. You know, in most of the cases, some baked goods made especially with baker's yeast, and as we heard before, with refined flour, have rather high glycemic index. This is a scale that has been done by the medical school in New England. What I want to show you is that through the sourdough fermentation, you can move the glycemic index of baked good made by wheat flour from high to moderate, and integrating this the sourdough fermentation with a low percentage of fiber, you can get products having low glycemic index. So the sourdough fermentation and the use of fibers or whole grains is the basic, is the fundament to get a low glycemic index also for these products. We did also comparing the sourdough fermentation with baker's yeast and human challenge regarding digestibility, summarizing the main outputs of this human challenge. The sourdough bread has a faster orocecal transit time, has a lower postprandial glycemia, and you can get a faster absorption of amino acids in blood plasma. So the conclusion is that the sourdough bread is more digestible than baker's yeast bread. Concluding, I'm living, as you heard in my introduction, in Italy. I'm working in Bolzano. I'm at the Neutech Park, and we have a pilot plant, 3 pilot plants for making baked goods. We are dedicated most of our research activity on bakery experience. What we are doing, we are preparing sourdough at laboratory scale, And we transfer, we are doing a technology transfer from lab to industrial scale, and we are promoting the use at artisanal and industrial level. I would like to thank you for your attention.